Next, we're going to look at the liver. So it's the largest visceral organ in the body. Weighs about 1.5 kilos in a man, and it lies in the right hypochondriatic and epigastric regions. It also extends to the left hypochondriatic and umbilical region, and it performs very essential metabolic and synthetic functions. The anatomy of the liver, a little bit further, it is wrapped in a tough fibrous capsule to help protect it. It's covered by the visceral peritoneum, and it's divided into two lobes. So here we have got a horizontal cross-section through the superior abdomen, and it's showing you that most of the abdomen there is taken up by the liver. We've got a small part of it being the stomach, a little bit of it being the spleen, but the majority of it is the liver. Here we are looking at the liver. You can see it's divided into two lobes, and just poking out underneath it is the gallbladder. Here's the posterior view, again the left and right lobes, and you can see more clearly the gallbladder. You can see also the hepatic portal vein and the hepatic artery. So these important veins and arteries supply the blood supply to the liver, and blood that comes immediately from the small and large intestine travel up through the liver before going back to the heart. So if we look at the liver lobules, the basic functional unit of the liver is called a lobule, and each lobe is divided by connective tissue and has about 100,000 liver lobules in it, each about 1 millimeter. So we're talking really small in diameter. And they have a hexagonal in cross-section, and they have 6 portal areas or triads, one at each corner of the lobe. So we're going to have a look at those a bit more closely. Here we have the lobes. So this is a single liver lobe that you're looking at, of course a diagram of it, and you can see here that we've got the sinusoid, which is the venous blood vessel, and you can see the central vein up the top. What I want to point out to you is the Kupffer cells. You can see here that they're attached to the inside of the blood vessels, and that's where they do their work in absorption. Here we have another one. Right, so you can imagine there would be hundreds of thousands of these through the whole liver. And the blue bits, of course, are the veins, the red colours are the artery, and the green bits are the bile duct. So, as we said before, the liver is involved in the manufacture of bile, and these are the collecting ducts. So the hepatocyte that we've just looked at adjusts the circulating levels of nutrients through the selective absorption and secretion. In a liver lobule from a series of irregular plates arranged like wheel spokes that we saw before. So here we've got, if we're looking at this central bit in here, then you've got all these bits on the outside which look like the spokes of a wheel. We've only got part of the picture, but of course it would go round. Many Kupffer cells, they're also known as stellate reticuloendothelial cells, are located in the sinusoidal lining. The sinusoidal lining is the blood vessel lining. As blood flows through the sinusoids, the hepatocytes absorb solutes from plasma and secrete materials such as plasma proteins. As I said previously, the liver secretes bile into a network of narrow channels called bile canaliculi, and between opposing membranes of the adjacent liver cells. So remember having a look at that picture in your anatomy and physiology book. You can see quite clearly that throughout the liver hepatocytes you've got these bile canaliculi which are absorbing bile and helping it to travel down to the common bile duct. So here we've got the posterior view of the liver and the presence of the bile duct and the gallbladder, where the bile is stored. And you can see also that where the actual common hepatic duct joins onto the common bile duct, and that common bile duct then travels down and joins onto the duodenum and is stimulated to release bile in the presence of digested chyme moving from the stomach. So the right and left hepatic ducts, they collect bile from the bile ducts of the liver lobes, and that unites to form the common hepatic duct, as I said before. The bile flows from the common hepatic duct to the common bile duct, which empties into the duodenal ampulla. The cystic duct leads to the gallbladder. The common bile duct is formed from a union of the cystic duct and the common hepatic duct, and it passes through the lesser omentum towards the stomach, and it penetrates the wall of the duodenum, where it meets the pancreatic duct at the duodenal ampulla. So what's the function of the liver? It's involved in metabolic regulation, hematological regulation, and the production of bile. Metabolic regulation. The liver regulates the composition of circulating blood, so it's involved in recycling old red blood cells. It breaks them down, and that's what gives feces that brownish colour. It's involved in nutrient metabolism. It's involved in waste product removal. It's involved in nutrient storage, so the liver can store excess glucose in the form of glycogen, which is then a form of fat, and it's also involved in drug inactivation. So if a person's liver is not working properly, then this will impact on the drug level within the body. So if they've got a liver that's only working at half the pace, that will impact on the amount of medication that person would receive, because that if it's working at half the pace, then the same dose of drug is going to take twice as long to be removed from the body. All of this needs to be taken into consideration when prescribing medication doses. So the composition of circulating blood. All blood leaving the absorptive surfaces of the digestive tract enter the hepatic portal system. So this is the venous system. It flows into the liver. The liver cells extract the nutrients or the toxins from the blood before they reach the systemic circulation through the hepatic veins. The liver removes and stores excess nutrients. It corrects nutrient deficiencies by mobilizing stored reserves or performing actual synthetic activities where it makes some things. Metabolic activities of the body, of the liver. It can metabolize carbohydrates, metabolize lipids and amino acids. It can be involved in waste product removal, vitamin and mineral storage, and, as we said before, drug inactivation. Hematological regulation. It's the largest blood reservoir in the body. So if you're looking after a patient who with a suspected traumatic injury, when you think about people in motor vehicle accidents, if they're sitting as the driver and they have their seatbelt, no, not the driver, the passenger who has a seatbelt coming from their left shoulder and buckling up on the right hand side, then if they're involved in a sudden impact, that sash from the seatbelt can be involved in lacerating the liver in an internal injury. So that's why we observe patients very closely for internal injuries after motor vehicle or high impact injuries of any sort. And it receives 25% of the cardiac output, so it has a very large blood supply. Functions of the hematological regulation. So phagocytosis, remember phagocytes are white blood cells, and antigen presentation. So the synthesis of plasma proteins, the removal of circulating hormones that are no longer needed, the removal of antibodies that are no longer needed, the removal or the storage of toxins, and the synthesis and the secretion of bile. So what is bile? it looks like it's green, which it is. Those of us who've had an infection of the intestinal tract may well have been involved in vomiting, and after we've brought up all of the food contents, the next bit that you start to bring up is sort of this greeny coloured fluid, and of course that is bile and mucus and saliva. So functions of bile: it's a dietary lipid that is not water soluble. It's involved in a mechanical process in the stomach, creates large drops. So the mechanical processing in the liver creates large drops containing lipids. So it breaks down the fat, but they're still too big to be properly absorbed. The pancreatic lipase is not a lipid soluble, so it interacts only on the surface of these droplets. So then comes bile. Bile salts break down these droplets in a form of what is called emulsification. And it increases the surface area of each of these lipid droplets so that they're available much smaller, so that the enzymes can actually get involved and break them down. So biles create tiny emulsion droplets which will be covered with bile salts to allow the enzymes to further digest the fat. The gallbladder itself is a hollow pear-shaped muscular sac. It stores and concentrates bile prior to the excretion into the small intestine. It's located in a fossa of the posterior surface of the liver of the right lobe, which we saw. The cystic duct extends from the gallbladder and it unites with the hepatic duct to form the common bile duct. Here we've got that representation again. You can see that it does look something like a pear, and you've got the cystic duct and you've got the hepatic duct joining together to form the common bile duct.